Hey everybody, and welcome to Unit 6, Lesson 1, Exploration 3. And today we're going to be taking on a big task, and we're going to be talking about DNA replication. And anytime you talk about replication or to replicate something, you're talking about copying it. And obviously when you copy something, the key is that it has to be absolutely identical. And you're not dealing with copying and having a little bit of differences. You're talking about copying and it has to be absolutely the same. So when we talk about replication, we talk about a process called semi-conservative. But what that's going to mean is we're going to use one side as a template to make the other side. So obviously DNA replication is the process which DNA is copied. So it's going to take place in the S phase, and that's why it's really important that we still understand this cell cycle, right? So interphase is kind of how we're setting up to divide. Then mitosis, we've talked about PMAT, prophase, metaphase, anaphase, and telophase, and then cytokinesis is the end. But now what we're going to do is we're going to go back to interphase, and we're talking right here. S phase, DNA is replicated. So before we just kind of skimmed over that and said, hey, by the way, here are the DNA copies. But now we're actually going to talk about how does it do it and what's the process in which we are going to use to copy our DNA. So as we scroll down, we're going to use what we call enzymes. And so enzymes are proteins, but what their deal is they have one job. Right? So enzymes are going to have one job, and we're going to use several enzymes. But anytime you hear an enzyme like DNA helicase, or you hear polymerase or ligase, all of these are talking about proteins that are going to do a job. So real quick, we're going to go ahead and hit play on this and take a look at it as we look through DNA replication. So here is our DNA. It's a double helix. You can see the two strands. Okay. So what's going to happen is they're going to pick a spot and it's going to start opening. And this is going to be a site. And this is what we call DNA helicase. And what's going to do is it's going to open. I'm going to pause it right here. It's going to open up the DNA. So we know DNA is double-stranded. This is going to be one strand and this is going to be another strand. And what's going to happen is we know that if this is an A, then the nucleotide that's going to be brought in on the other side is going to be a T. Well, the good news is that over here we have the A and then we're adding in the T. So that's why we're going to end up getting two exact copies, is because we know that A and T only go together and C and G only go together. But what you're going to see is these what we call free-floating nucleotides, and they're going to be bringing them in on the opening side on both sides. So we'll go ahead and hit play, and here they come. As you can see, they're bringing it in. You can see that this one's running right behind this enzyme. However, these are actually working backwards. And you can see that they kind of get thrown in backwards like that. And that's called the lagging strand. And we're going to talk about that as we get going. So one strand is going to be made continuous. This is called the leading strand. And then the one that's going to be made backwards is going to be called the lagging strand. These little things are called Okazaki fragments. We're going to talk about that. And then this enzyme right here is DNA ligase, and it's going to link everything together. So it says right here, some enzymes start the process by breaking the weak hydrogen bonds down the middle. Remember, the hydrogen bonds are the ones between A and T. But what's going to happen is we're going to get a series of enzymes called DNA polymerase. And DNA polymerase's job is to bring in nucleotides and bond them so that we get an identical strand of DNA. So it's going to use both sides as a template, and we're going to bring them in. So step number one right here is going to be helicase. So helicase is an enzyme that's going to unwind and then open the DNA. So all of these are little hydrogen bonds between the A and T's and C's and G's. And what's going to happen is helicase is going to open it. We're going to get one going this way, but we're also going to get one here going the other way. And what it's going to do is it's going to start opening up that DNA. So then that way we can start bringing in free-floating nucleotides on this side and on this side. Okay, and what you'll notice is that yellow and purple are going to go together. So what's going to happen is when polymerase, if we have yellow, it's going to bring in a purple. But we also know down here that we're going to have a purple, and it's going to bring in a yellow. So right here, we're going to have the exact same match of yellow, purple, yellow, purple. And that's really important because it's going to do that all the way down, and that's how we're going to get an exact copy. So as we scroll down, it talks about nucleotide pairing, and that's what I just talked about. An enzyme called primase is going to make a little RNA primer. Okay, that's kind of like the beginning, right? So if you think about a zipper, okay, a zipper has that little spot down the bottom where your zipper, where it actually inserts into the zipper, and then the teeth engage. So that little spot is kind of like a primer. We have to set this little spot before the polymerase can start. Okay, so here's a graphic down here. So here's our little primer. And as we can see, DNA polymerase is going to work this way. And as it brings it in, it's going to bring in the matching nucleotide. Okay, this is called the leading strand because it's going to be made continuous. Now, if you look down here, we're going to have the lagging strand because the lagging strand is going to work backwards. And this one, this polymerase is actually working this way. So as we scroll down a little bit, here is a picture of the lagging strand. So the lagging strand, you can see the arrows, they're working backwards. And this one has locked on right here and it's working backwards. And as helicase opens up the DNA, there's another one that's going to lock on and work backwards. And here it is kind of both together. So we're going to have one going this way and we're going to have one going this way. And as you know, DNA is anti-parallel, right? Remember we talked about how there's a direction to the DNA. So this one's going that way and this one's going the opposite direction, okay? As we scroll down here, so here what we have is, at the end, what we're going to end up is 2 exact copies of DNA. If there's green and red right here, there's going to be green and red right here, and yellow and purple and yellow and purple, and so on. They're going to be absolutely 100% identical. And this is how we go from one cell at birth all the way to over 100 trillion cells that all are going to have the exact same DNA in every single nucleus. Okay? So it talks about fast and accurate replication. This is how it actually happens. So instead of just one helicase, there's actually going to be anywhere from 3 to 4 to 5, depending on the chromosome, and they're going to start working out from the middle. And so what's going to happen is we may stagger them out like this, but then as they go through replication, they're going to get bigger. We call these bubbles. And then they're going to get bigger, and then sooner or later they touch, and then they leave, and there's our 2 strands of DNA. Okay, so we're going to take an extra day in class to talk about this and kind of continue our process of going over DNA and how it replicates. But this is a good start. I'm glad you joined me and can't wait to see you all in class.